Thank you. So thank you all so much for coming today. My name is Alex Harkess. I'm currently a postdoc with Blake Myers at the Donald Danforth Plant Science Center in St. Louis, but I'm going to be telling you about some work with Jim Liebensmack at the University of Georgia today. And this is specifically about the evolution of a sex chromosome pair in what you might think is a seemingly ordinary grocery store vegetable, but I'm going to change your mind today about that. So the overwhelming majority of flowering plant species are hermaphrodites, meaning that within every single flower, there are both boy and girl parts. But in a relatively small fraction of flowering plant species, we see dioecy, or the separation of male and female organs onto entirely separate plants. And dioecy is a rather peculiar condition across the angiosperms, in that only 6 to 9% of flowering plant species might be dioecious. And well, dioecy is rather fascinating because it's evolved hundreds of independent times across the angiosperm phylogeny. And in many cases, dioecy can be accompanied by the evolution of a sex chromosome, an XY or a ZW system that will actively control the determination of gender in a species. But a very obvious question might be, what are the genetic mechanisms that drive the conversion from a regular autosomal pair in our hermaphroditic species to an XY or a ZW sex chromosome system in a dioecious species? Well, back in the late 70s, Deborah and Brian Charlesworth formalized a few hypotheses about the conversion from an autosome to a sex chromosome. And they hypothesized that this transition might actually be quite simple, that you might only just need 2 genes in order to convert a hermaphroditic species into a dioecious one with an XY sex chromosome system. They hypothesized that you need 2 genes linked together in perfect non-recombination on a neo-Y chromosome. One of these genes must somehow suppress female organogenesis, and it must be perfectly linked to another gene that promotes the formation of male organs. And this is a really elegant and simple hypothesis, and it makes a lot of sense for us to grasp. But even though we've been sequencing and assembling multiple dioecious plant genomes over the last 5 and 10 years, no one has actually been able to show that this hypothesis is how hermaphroditic species convert to dioecious ones. Well, today I'm going to convince you that asparagus, what you see in the grocery store, is an excellent model system for studying the conversion from autosomes to sex chromosomes during the evolution of dioecy. Well, chloroplast phylogenies in the asparagus genus reveal that there might be 1 or potentially 2 origins of dioecy and a sex chromosome. So about 1/3 of all asparagus species are dioecious, the other 2/3 are hermaphroditic. And so what you see in the grocery store, Asparagus officinalis, is a classic dioecious species. We have very beautiful separate male and female organs. If you're not a botanist, these are anthers right here, and here we've got pistils right here, and the stigmatic surface where pollen will land on. Now, developmentally, all asparagus flower buds, whether you are a male with X and Y chromosome or a female with 2 X chromosomes, they initiate as hermaphrodites. All flower buds do. Early on in XY male development on the left, though, the styler 2, which would come out right here, begins to form, it flops over, it falls off, and you're left with a functionally male flower. On the other hand, in XX females, anthers, the male parts, almost nearly fully form until the very last second, right before pollen microsporogenesis occurs. The tapetal layer, this layer that surrounds developing pollen grains and is incredibly necessary for their development, begins to crinkle up and crush, and then the anthers fall off, and you're left with a functionally female plant. So we've got 2 temporally separate sterility events, a male sterility and a female sterility event. And so this might lead us to believe that we could potentially have a 2-gene sex determination system in asparagus. But to really answer this question, we need a genome sequence, and we need a genome assembly. Well, asparagus is really excellent to work with from a genetic standpoint because we're able to produce doubled haploids. So the asparagus genome is about 1.3 gigabases. It's pretty large. But we can do anther culture. We can diploidize a pollen grain, basically, to produce an entirely homozygous genome. And so we've sequenced and assembled this perfectly homozygous YY supermale genome. It's got 2 Y chromosomes, using a combination of short insert and large insert 20 and 40 kb mate pair libraries with Illumina sequencing, PacBio sequencing, and we generated an initial assembly that was pretty decent with a scaffold N50 roughly about 25 kb. But then we've applied some interesting other techniques as well to improve this genome assembly, including genetic mapping and optical mapping. So since I told you that we can make these doubled haploid individuals, we can actually produce a population of doubled haploid individuals derived from a single XY male. And so of this population, we have XX and YY individuals that we can use for linkage mapping. And so with a 72-member population of perfectly homozygous individuals, we were able to construct a pretty decent genetic map for which we could use to anchor and order and orient a lot of our genome onto pseudomolecules. And so with a 3.2 million SNP map, we had pretty good resolution to do that. Lastly, we used this technology called optical mapping through bio-nanogenomics to further scaffold and order and orient our assembly. Optical mapping is where you can stretch out ultra-long pieces of DNA, nick them with a single-stranded restriction endonuclease that fluoresces, and then you run these really straight DNA molecules through a camera, so you can get physical distances between these fluorescent marks. It's basically like back fingerprinting on a huge scale. So then you can match up these fingerprints from raw DNA onto fingerprints from your genome assembly, and you can incredibly scaffold your assembly to up to 10-fold increases of what we're seeing. And so all in all, what we ended up with is a pretty incredible genome assembly for garden asparagus, anchored and ordered and oriented onto 10 pseudomolecules. The color's a little washed out here. When we look at the gene annotations, we see a pretty typical plant genome here with an increase in genes out near the ends of chromosomes and a high frequency of long LTR retrotransposons near the pericentromeres and the centromeres. But the obvious question is, which one of these is the sex chromosome, and where is this sex determination region that might be harboring these sex determination genes? And so previously in the past, people had identified sex-specific markers, but they weren't great. But we were able to use those markers to identify about an 862 kb contiguous region on this young Y chromosome that is largely hemizygous. So it's missing from the X chromosome and only specific on the Y. And so this is a really obvious place to search for these sex determination genes. And so when we look at our gene annotations across this entire region, we really only find 7 gene annotations across this 862 kb region on the Y chromosome. There's a few extra genes that we weren't able to order and orient into this 862 kb, but we feel very confident in our assembly because we were able to line up the optical map against our sequence assembly, and they are largely matching each other. So again, the utility of optical maps to span difficult regions in plant or animal genomes can't be understated here. And so the obvious question is, well, what are the genes in this region? There's only 7 of them. And so when we look at these gene annotations, only 5 of them really have hits to the UniProt KB database. And the first thing that we really thought about was, well, are we sure that this region, this 862 kb region, is the sex determination region? Are we going to find sex genes here? So we were able to leverage gamma irradiation, reverse genetics. So we were able to blast a bunch of XY males with gamma rays to randomly knock out large pieces of DNA, and we knocked out this entire region. And so what we got was a male to female conversion. And to do that, there has to be at least 2 sex determining genes, and that fits our model. So talking about these annotations, I'm going to point you to this transcription factor first, a MIB35 transcription factor, otherwise known as defective in tapetum development and function 1. Now remember, in asparagus females, they produce anthers until the very last second. Right before pollen's about to be formed, the tapetal layer in female anthers crushes up and falls off. Now this gene, TDF1, is only on the Y chromosome. It's missing from females. And so we can leverage comparative genomics a little bit here by looking at the Arabidopsis thaliana tDNA knockouts, and what we see is a nearly identical phenotype. That Arabidopsis knockouts of this TDF1 gene, which is single copy in both asparagus and Arabidopsis, they don't produce pollen, but the female organs are left entirely unaffected. And that's exactly what we see in asparagus as well. And so this is a pretty incredible candidate gene. The phenotypes match. But of course, in the context of our model, we still need another gene linked to this one that would suppress the formation of female organs if we're fitting the Charlesworth model. So how do we go about identifying a gene that will dominantly suppress female organogenesis in a male plant? Again, we took a gamma irradiation approach where we gamma irradiated nearly 40,000 XY male seeds. And what we were looking for is a conversion from a male to a hermaphrodite via the knockout of a female suppression gene. And so after irradiating 40,000 of these XY male seeds, our collaborators in the Netherlands found a single mutant. And this mutant has a functioning stigmatic surface here. Here's the girl part, basically, if you're not a botanist. And the male parts are left entirely unaffected. And so we were able to produce hermaphrodites in the field through this irradiation technique. And when we go back and we resequence these individuals just using whole genome shotgun resequencing, in this one individual that we found out in the field, there's a single gene deletion, actually a single exon deletion across the entire genome. And this single exon deletion occurs in one Y-linked gene in this 862 kb region. This gene really has only one annotation, and it's a domain of unknown function. It was a disheartening annotation to see because no one knows what this gene does necessarily. And of course, this gene is also missing entirely in females, which again makes our model, or matches our model. And we're also pretty confident about this gene because when we look at this TDF1 gene as well, we don't see any effect in these gamma mutants. And so again, we think we've got a single exon gamma irradiation deletion, which led a male to convert into a hermaphrodite. At about the same time, our collaborators in Italy have a glass house with a bunch of asparagus plants in it, and they have known the gender of all these plants for a long time. And so one year they noticed that a plant that had typically always produced male flowers was producing hermaphroditic flowers. It was a spontaneous mutant. And of course, that's odd. So they sent us this plant. They did a little bit of RNA-seq as well. And what we see is that there's a single base pair deletion in exon 2 of this same gene, the same female suppression gene. And so we've got two independently derived knockouts, basically. This single base pair deletion leads to a premature stop in what we assume to be a non-functional protein. And so this all comes back to the question, who's there? Do two sex determination genes exist on the Y chromosome? Were Deborah and Brian Charlesworth potentially right, at least in one plant species? Well, we feel incredibly confident that we have identified a dominant female suppression gene. The use of reverse genetics and the identification of two independently derived male to hermaphrodite mutants is incredibly convincing. But have we identified a male promoter? Is this TDF1 tapetum gene a male promoter on the Y chromosome? Well, it seems very likely. But of course, we need to be our harshest critics. And without 4 genetic knockouts, say through CRISPR-Cas, we can't definitively say that this is the male promoting gene. We need to knock out all 7 of these genes in this non-recombining region. Further, we can leverage comparative genomics as well by sequencing and assembling additional dioecious species, more X and Y chromosomes, as well as hermaphroditic species with an autosome to identify if there are any changes in this small region. So with that, this has been an intensely collaborative effort across many universities, across the world really. But I would like to give a special thanks to John Bowers at the University of Georgia, who did much of the genetic mapping, Rajai Appalayam, folks at the University of Reggio Calabria in Italy, who identified a spontaneous mutant in their glasshouse. And in particular, this one breeding company, Lim Group, over in the Netherlands, was incredibly useful in identifying these gamma irradiation knockouts, because I do not have the power myself to phenotype 40,000 plants in the field. So with that, I'd like to take any questions, and thank you all so much.